starting this. So hi everyone, welcome to this week's biology seminar. Today we have Professor Jorge Rodriguez. He's from Khalifa University. Just to give you a little bit about his background, he's mostly an engineer, actually, environmental engineer, but he does a lot of work on microbial systems and modeling, and you'll see some really interesting stuff that he's doing in his talk about modeling metabolic pathways. So Jorge got his Bachelor's of Engineering from Universidad de Santiago de Compostela in Spain. And then he moved on and got a Master's in Engineering from the same university. And then he got his PhD as well from there in environmental engineering back in 2006. After that, he's been in several positions. He moved quite a lot actually. Postdoc fellow in Australia in University of Queensland, and then moved to University of South Wales for a bit of time. And then he came here to the UAE, where he became an assistant professor in what was Masdar Institute of Science and Technology back there in Abu Dhabi, which is now Khalifa University, and he's now an associate professor in chemical engineering at Khalifa University. So this, and just to add, Jorge has also a lot of kind of recognitions and awards. He's a fellow of the International Water Association. He's also on the associate editor of a number of journals, including Frontiers in Microbiology and Frontiers in Chemical Engineering, among others. I'm not going to just keep going. I'll just stop right here, and I will leave the floor to Jorge. I'm happy to hear you.
Thanks, Shadi. Thanks for the introduction. It's really great to be again talking for events that you are organizing. So it's really nice to participate with NYU Abu Dhabi events. Let me know if my screen or sound doesn't work properly, just so we could correct it. So, well, when you guys invited me today, I asked back a little bit what kind of audience was going to be today in the seminar. And then from the feedback you guys gave me, I decided to do the talk in like basically two parts. Okay, we're going to be talking about bioenergetics and thermodynamics a little bit, and how we can apply that to microbial systems. That will be a little bit like an introduction for some, a refresher for others, and it will put in place all the concepts we are going to need to understand better the research work I will present in the second part. This work is a work in collaboration with one of my PhD graduates, Mauricio Paton, who's still as well with us as a postdoc researcher. So we put a title a little bit just to catch interest: microbial prospecting using bioenergetics and pathway modeling. Why this prospecting? Because I think it could be kind of a theoretical prospecting that I think could come out of this work. And then the idea is whether or not we can predict pathways, community function, and growth yields based on some calculations, some thermodynamic calculations. Okay, so my idea is to start this first half of the talk by introducing some bioenergetics in microbial systems, and how do we actually calculate things? How can we calculate things for these microbial systems? Initially, we can just start talking about energy. Right? If we are looking at studying bioenergetics, well, that's energetics. So what is the energy? Why do we need energy? Why do cells require energy? We know that living matter is a highly organized, we are highly organized systems. Cells are highly organized systems. Therefore, we need to maintain that order. So cells are going to need energy to maintain order, to maintain entropy at a low level inside the cells. Obviously, living things have to grow, and sometimes they also have to move and do all the functions. So that's the main reason we know that energy is needed in cells. What we know is for chemotrophic organisms, those that are doing that are getting their energy from from some chemical transformations, the catabolism is the source of the energy. So we're going to have some energy levels here in the reactants, and there will be some catabolism taking place, and then we have some products. By necessity, the energy content of those products is going to have to be less than the energy content of the reactants, because we are harvesting, the cell is going to be harvesting some of that difference between the two. Okay, so this is the main initial idea I wanted just to introduce in your mind. Obviously, we know that biology does chemistry through a catalyst, very specific catalysts, which are enzymes. And these enzymes, what they do is they lower this activation energies required. So we start here, we need to go up the hill and go to the lower level of energy in the products. So obviously with enzymes, we know this hill is going to be less high. So it's going to make the reactions go faster. But we're not going to talk about the rate of reaction today. I'm going to talk about only the energy and the efficiency of the reactions. So, okay, we have our substrates, our products. So some microorganisms might be living on that chemical transformation. And there is energy at the beginning and energy at the end. And this difference between those two products and substrates is going to give some energy exchange, some energy that was made available. So a cell needs to be able to harvest that energy. So, and we know this is biology, from basic biology, the mechanisms for energy conservation in the cells are these two mechanisms. You can either have a reaction which is stoichiometrically coupled to an ADP reaction. So you add the phosphate through a stoichiometric step in a particular reaction. So you make substrate level phosphorylation. This is one way of conserving energy. For that to happen, you need to have a reaction that is having sufficient energy difference to actually put that phosphate in the ATP. So it needs to have an available amount of energy bigger than the delta, the Gibbs energy change, or the energy quantum of the ATP to ADP reaction. We know also that when that's not available, we can still have energy conservation. We know that the cells can actually use the proton motive force. There are reactions that might be coupled. They might have less energy than the amount for ATP. So what they can do is to just pump a proton outside the membrane and generate that proton pump, which is a smaller quantum of energy than the ATP itself. And then we know the mechanism of the ATP synthase in the membrane. So those protons can come back. So between 3 and 4 protons, depending on the specific case, they will be producing enough energy to load up another ATP from an ADP. So that's one way of using smaller quanta of energy when the reactions don't give us the big quantum of energy for ATP substrate phosphorylation. We can still conserve energy with smaller quanta as proton translocations. So these are the two mechanisms we know life conserve energy. And then we can always look at the how this is all organized. We have substrates becoming products, they generate that energy. Part of that energy will be conserved in form of ATP, and that ATP can be used for maintenance, and they can be used for anabolism to make to create biomass growth based on a given carbon source or nitrogen source to build new biomass. We have, typically the values that we know, the ATP, how much energy is in ATP is about 50 to 70 kilojoules per mole, depending on the concentrations and conditions. We're going to be talking about these things a little bit in a moment. So how do we quantify that energy? What magnitude are we looking at in chemistry when we look at reactions, whether a reaction can proceed or not, is the Gibbs free energy. The Gibbs free energy is basically telling us whether, if I have a particular situation and I have some chemicals and the chemicals could be transformed into some products, is this actually possible? Is that going to happen? Okay. And then if it's possible, then maybe there is some microorganism that is going to be able to do it. Okay. But if it's not possible because of the loss of thermodynamics, then we know that there is not going to be anybody or any microorganism doing it. So what tells us whether a reaction can actually proceed is, as a requirement, is that the amount of Gibbs free energy available decreases. Okay. What is the Gibbs free energy? The Gibbs free energy is actually the amount of energy available to do work in a system. And work is including chemical work, chemical rearrangement. So if you reach a point in which there is no more energy exchange, whether you go forwards or backwards, then we reach what's so-called the equilibrium, because there is no more energy available to do any work in any of the two directions. So we reach equilibrium. When the energy exchange is negative, then the reaction is feasible, meaning what's called exergonic or feasible, meaning it could happen. In principle, there is going to be energy available to be released if the reaction happens. It doesn't mean it will happen very fast, depends on other things like, do we have a catalyst? Do we have somebody doing it? Is it a very big hill that we need to climb? That's another story, but in principle, the reaction can happen. If the reaction has a positive exchange of Gibbs energy, then we know that it's not feasible, right? It's an uphill all the way net, meaning it's not going to happen. Okay, and then the Gibbs energy has actually two terms. We might be familiar with enthalpy, you know, exothermic, endothermic reactions. You probably heard that before. Exothermic reactions, they release heat. Endothermic reactions, they absorb heat. And then there is a second term here in the Gibbs energy, which is the entropy, okay, which is the talking about how you are creating more chaos or disorder, or you are actually creating a more organized system. Okay, that also weights. And here there's some examples. Actually, if we imagine that we start here at 0 and we have certain reaction, if we need this amount of energy to sustain life, for example, then if we look at things like the methanogenesis from acetate, which is a microbial process, which converts one acetate into one methane and one CO2, which it means 2 molecules. Will that happen? We know it happens. So how does it happen? If we look at the Gibbs energy, it's actually an endothermic reaction. Okay, but thanks to the increase in entropy, the reaction actually runs because you're creating 2 molecules out of 1, so you're creating more disorder from the single molecule that you had at the beginning. So in that case, it happens. Other fermentations, similar way. Fermentations typically increase the number of molecules. You can have hydrogen as a product, CO2, and other smaller things from a bigger glucose, for example. And then they might also be exothermic if both point in the same direction, so both add up to the negative. When we look, for example, at aerobic respiration, this is a high energy. This is like a combustion, right? So glucose plus oxygen, this is a lot of energy into that. We know it's very exothermic, so we have plenty of enthalpy pushing and also a little bit of entropy. We know that has a huge amount of energy. We'll talk about it later. And then other systems, like, for example, making methane out of CO2 and hydrogen, there you reduce the number of molecules, so you have less molecules. So your entropy pushes in the opposite direction. So you're creating order, but it's very exothermic. So you still win and you get a negative net. So these are examples how these different elements contribute to whether or not a reaction can run. So how do we calculate these things? If I have, let's say, the example of glucose oxidation, I go to the books of thermodynamic properties and I get the values of Gibbs energy of formation for glucose, oxygen, water, and CO2. I plug them in, and then I do some calculations, right? So times 6, times 6, times 6, and times 1, and then I add them together, and then I get a number. The difference in energy from the products minus the substrates is minus 2,880 kilojoules per mole. That's the energy I have when I do glucose with oxygen, which is what our cells do, for example, and many heterotrophic organisms do as well. So that's one case. But what if we do something else? What if we do glucose to methane and CO2, for example? This is the complete methanogenesis, all the steps of methanogenesis. How much energy is available if we convert glucose into methane and CO2? You do the calculations in the same way. We take the methane Gibbs energy formation and the others, and we calculate it, and we look at this number. Now it's minus 428, okay? Which tells us immediately, as we knew it, that if we do an aerobic oxidation of sugar or glucose, we have lots of energy available in principle. If we do fermentations, we're going to have less energy available because we don't have a strong electron acceptor, so the energy exchange is going to be much smaller. This is all nice. We did some calculations, but there is a bit of a problem additional here, because normally when we go in the books, what we find in the books is the values of Gibbs energies in standard conditions, which means 25 degrees, pH 7 if standard biological, and then 1 molar concentration and 1 bar for gases, which is, you could say, okay, yeah, this is standard conditions. It might be good enough for me. The problem is that most of the time, or actually always, we are never going to be in any real system, in any biological system, in standard conditions. I mean, where do we have 1 molar concentration of anything? It's very, very rare, very unlikely, right? So we need to account for the effect of not being in standard conditions. Okay, and this is normally this Gibbs formula that you might have seen in the undergrad chemistry or thermodynamics, which basically tells me the actual Gibbs energy exchange of my reaction in my conditions is the standard one that I take from those books, plus some deviation from that standard. And that deviation we calculated as RT, logarithm of the products to the power of the stoichiometry multiplied by each other, and then the substrate multiplied by each other. What does it mean, this reaction? If we accumulate a lot of products, then this becomes more positive, okay? And then the reaction becomes less negative, less favorable, okay? Is that important? So we can actually do some calculations for some more realistic cases, right? So let's imagine, for example, if I'm trying to calculate the actual energy available in propionate oxidation in a methanogenic environment. What happens there? Well, we have propionate, typically 1 millimolar, going into acetate, let's say another millimolar, CO2, let's pick 10 millimolar CO2, and then we have hydrogen production, but the hydrogen pressure is not going to be 1 bar. Of course not. It's going to be around 5 Pascal, for example, typical in a methanogenic environment. We plug in those numbers. We get from the books the actual standard value, which is as we did before, and it's actually positive. So it would never run. This would never run in standard conditions. But now we modify and we apply our concentration. So we put, this is our standard value that we just calculated. If you apply the concentrations of substrates and products, then you recalculate it, and then voila. Now there is actually some energy available to do this process. We know this process runs, so we know that somehow it needs to be running in conditions that is actually favorable. It's actually 18 kilojoules per mole, which is tiny, but it still is already negative. And then we're going to be talking a little bit later whether, I mean, in what pathways, how can microorganisms perhaps live on that little amount of energy with the propionate oxidation. Okay, so actually concentration changes actually do matter, but do matter in systems which are close to equilibrium, which is low energy systems. We saw the glucose plus oxygen had 2,800 kilojoules per mole. So we're never going to really stop that guy from running, no matter how many things we accumulate in the products. But if we are at a small amount of energy, like we just saw just now for the propionate oxidation, if things accumulate, and remember, some of the products might be three molecules or 4, so they are to the power of 3 to the power of 4. And if you make a ratio of 10 to 1, 100 to 1, 1,000 to 1, this plot actually shows you the impact in the energy part of it, right? So let's pretty much every order of magnitude you increase this whole term here is 2.3 kilojoules or positive on the side. And then if you put a lot of products at higher concentrations, a lot of substrates at smaller concentration, and they are elevated to some powers, this can become actually significant, like it did here, as you saw in this previous one, right? We moved from 76 to 18, but we have hydrogen to the power of 3, and look at the value was 5 Pascal instead of 100,000, so it was a big difference. These are the guys where actually 1 millimolar is the same, and 10 millimolar with respect to 1 molar in standard conditions is actually another factor of 2 or 3 orders of magnitude. So easily these added up the energy required to bring this 76 to minus 18. So it's possible, but that's not going to happen in aerobic systems at 2,800. But it will happen at these guys with a few tenths of kilojoules. So this is actually relevant. These calculations are relevant in energy limited systems. And this is nice because then we can actually calculate sort of energy niches that can be created in particular systems. I have this example here. I prepared some time ago for an article. If we look at, for example, a reaction, the blue one, butyrate to acetate and hydrogen. Okay, if we look at the standard, it's very positive, not very likely to happen. And then if we look at the alternative butyrate reduction with hydrogen into butanol, the green one, in principle looks standard. It looks favorable in standard conditions, but we don't care about standard conditions really, because we're never at standard conditions, and the numbers are small. So we know these numbers can actually become positive or negative depending on the situation. So what we can do is start playing. I can fix the concentrations of butyrate, acetate, and butanol, and I can play, for example, with hydrogen and pH, which are actually products in the blue one and reactants in the green one. And you see how the Gibbs energy, this is minus the Gibbs energy, so actually the positive is the negative. You see the reaction becomes more favorable. For example, the green one becomes more favorable as we have higher hydrogen. It makes sense because it's actually a reactant, but that has the opposite effect on the blue one, on the oxidation, because hydrogen is a product. So the more we move in this direction, the Gibbs energy eventually is going to cross the black, the floor into positive Gibbs energy. So it's never going to run. So these actually, and the same with pH, right? It has some effect. So we can actually make these plots and create this niche in which both of them can actually live. But if we are outside those ranges, actually only one of the reactions might happen. Okay, and then you can imagine a bacteria in which you put it in a batch system, in a bottle closed system, and then initially you start in point A. This bacteria starts making more hydrogen and PA and protons, so it pushes the hydrogen pressure a little bit that way and the pH a little bit this way, and then it suddenly enables the other guys to do the work, right? So it's a bit like a Winogradsky kind of story you can imagine. And you can do this little calculations with thermodynamics. Here I have another example. Imagine we do electrochemically, exoelectrochemical, let's say in an anode, butyric oxidation to an electrode, and we play with the voltage of the electrode. Now I froze the pH in this one, so I move the hydrogen and the electrode. So you can actually even calculate the feasibility of the reaction based on the voltage of the electrode. That was for another example in which we can see how the energetics of an electrode could also be could be brought into these calculations. So that's a little bit the initial introduction of these concepts that I want to put in your mind before talking about our research. At the end, I like this quote from Albert Szent-Györgyi that looks at he says life is nothing but an electron looking for a place to rest. So we can look at that from that perspective, right? That electrons are trying to just go at the lower level of energy, and then if some life can actually harvest some energy on the way, then is what allows us to stay alive and what allows the cell to stay alive. In an example is anaerobic digestion. Anaerobic digestion, you start with organic matter, and it's a fermentation, and then you get to the latest stages in which you have the methanogenesis, and then we wonder, okay, why is glucose becoming methane? I mean, what's the point? What is it going to methane eventually if you leave it long enough time in anaerobic conditions, no strong electron acceptor or anything? Because the energy content per electron decreases, so electrons actually are moving downhill to methane. So you start, how much energy is in an electron in glucose is 41 kilojoules. But in methane, you have different intermediates, ethanol, acetate, propionate, butyric, valerate, and then eventually methane goes there. So it's actually the energy is driving the energy direction. The energy arrow is pointing the direction to which things go. So we're not going to get glucose from methane and CO2 together. We're going to go the other way. So that's a nice way of looking at that. Okay, so I hope that puts some introduction, some concepts, as a refresher for some of you. So many of you might already knew this already because it's actually kind of undergrad, bioenergetics and thermodynamics. Now, what I want to do in the second part of the talk is present you some of the work we did for to try to theoretically find out what microorganisms should be doing to be the most efficient to achieve the highest yield in a particular environment. So the title here could be, can we determine the most energy yielding pathways in a microbial environment and their associated growth yields? The idea is you have an environment which we don't know, of course, we don't know what is happening there, everything in all details, but can we just combine all the biochemistry we know that is being seen in terms of reactions and enzymes that are available, together with these rules of energetics, and put it all together and see what is the best way for a microorganism to do this. And remember, if we're looking at a case in which energy is extremely limited, like the case of propionate oxidation, which I was showing before and I will show now, this is a very powerful selective force. So only those organisms that are extremely efficient, that have extremely efficient metabolisms, are going to be able to survive. If you are living out of nothing, you cannot really waste anything. So this is the actual case here. So in this work, we're going to see whether we can actually find out which are the best ways. And then this creates the hypothesis of those pathways being the predominant pathways in a particular system, which is interesting because then we'll be able to actually look for those pathways. We can target functional genomics, or we target other experiments to actually verify those hypotheses. And this is where we think is, we call it a little bit prospecting, because we could be prospecting theoretically and then experimentally a little bit. So what we try to do in this work was to create this tool to evaluate lots of possible pathway combinations. And that's what we mean by lots. I'm going to show you in a minute what all these variations are, because it's not only the main pathways that we know, but sometimes there are options of which electron carriers are we using. Are we translocating protons here? Are we translocating protons there? How many? All those things. So we wanted to actually evaluate all the possible combinations, right? So that was the objective. And we wanted to apply this for a particular case of propionate oxidation, because it's a very extreme case of energy tightness. There's almost nothing available for them to leave. So this is a perfect case for us. And then we also applied it for the syntrophic case, in which we have the propionate oxidation making hydrogen, and then we have a hydrogenotropic methanogens taking that hydrogen, and both have to live out of that little thing. they both have to find the equilibrium point in which they both can sustain life. And then we work a little bit on that. So what we did first, okay, how can you do propionate oxidation to acetate? So let's look in the books and see what are the main pathways. So we did an inventory of pathways. So we found this one, propionate to acetate this way, succinyl, methylmalonyl, blah, blah, succinate. This is one option, okay? And then we found variations of this one with a little cycle here, with a little cycle there. Another option is going through the lactate pathway, okay, the lactoyl-CoA and so on. Another option was this family of pathway, which is through hydroxypropionyl-CoA, which is another option. And then we also took into account this micella pathway, which is a slight variation of that stoichiometry. So these are the ones which we found in the literature, which are kind of the known possibilities in terms of main pathways to go from propionate to acetate and hydrogen to do the oxidation. Great. So what we do is we take all these steps, the individual steps of those reactions, when we label them here with numbers. So we have 25 possible reactions, which are each one of those little arrows here, which will be one of those steps. So the combinations of those, so we have these groups of reactions I show you now, number 1, 2, 3, 4, 5, 6. These were the big families that I showed you just now, okay, in the slides. So you can place in order which reaction, which ones are actually participating. So this is the first, the second, the third, fourth, 5, 6. So the order is written there. Very good. But then the variations come into place because now we have the possibility of using certain electron carriers, maybe FAD or NAD or a ferredoxin here or an NAD or an NADP, and those have different energy levels. So we wanted to try all of them in the sense that we have these 6 possible combinations of 7 families of reactions. But then I want to try also all possible combinations of feasible electron carriers because we don't really know, it's not really set in stone that this must be this one and this must be this one. So let's allow for all the possibilities. So that's the kind of level 2 of combinations. And then we added a second level, which is there are certain sites in which we know that protons can be translocated. And this is very relevant because this is where the energy is conserved. We have obviously ATP by subcellular phosphorylation in some of these reactions. Some of these reactions are coupled to ATP formation. They are labeled here for consumption. But now we know that other reactions can also be coupled to proton translocations, which is also energy meaningful in terms of a third of an ATP or a quarter of an ATP, depending on the value we used. And then what we did is, okay, where these protons can be translocated and how many protons? Well, we don't know. So we're going to cover a range. So we have like between -1 and +1, maybe, including 0, or between -2 and +1, between -2 and 1, or 0 or 2. So we give a range of possibilities. So when we actually do all the combinations of these, things get actually very quickly, the combinatory explodes because you have 6 and then all the combinations of carriers and then all the combinations of proton translocations, all possible ones. We ended up with like 500,000 combinations, which immediately tells us that we're not going to be doing this by hand. And then the point of the work was actually to develop a method to do this automatically. Okay. So what are we doing with this? We have this 500,000 possible ways to go from propionate to acetate, harvesting some energy, theoretically possible in the sense that are biochemically possible. But then this is not the only requirement for a reaction to be possible. For a reaction to be possible, we need to check other things as well. So a reaction can run only, we saw it before, when the Gibbs energy exchange is negative, okay, or almost 0, but it needs to be a little bit negative. So what we do is we're gonna check reactions are going to be feasible only if every step of the pathway, every single step of the pathway, with all the combinations we put in, is going to have a negative or a very tiny negative delta G at least, but a non-positive for sure. Okay. This is not the only constraint. We also know that the metabolite concentrations, you remember we saw it, that concentrations can change this Gibbs energy. So if we are in trouble, maybe in one reaction, we can lower the product concentration. The cell can lower the product concentration of reaction number 1 to make it possible. And then reaction number 2 is going to be a little bit more difficult, but maybe it works because you have a higher energy. You never know. What we know is that we cannot have any random values of intermediate metabolites because things cannot be, it's unreasonable to have in the cell more than 10 to the -2 molar. This is the limits we put. And then it's also unreasonable to have less than micromolar, or at least much less, Why is that? Because actually the volume of a cell is actually quite small. So when we have less than micromolar, we very quickly get into numbers of molecules that we can count with our fingers, which is somehow going outside the chemistry, right? So these are the limits we put in there. So that's fine. If I manage to get all the steps of my reaction with a negative and non-positive Gibbs energy exchange and all the internal metabolites concentrations, I manage to keep them between those two values, then my reaction possibility, my reaction variant is actually feasible. And then I can count how much energy I'm getting out of it. How do you count it? The ATP translocation, the ATP in the table I showed you before, number of ATPs, plus the number of protons that I have translocated in net. And then I can come up with a value of yield of ATP in that particular reaction of my 500,000. Okay, and then you try this automatically for the 500,000 reactions, and then the majority of them are actually not feasible right away. They don't work. And from the feasible ones, then there will be some of them that have a negative energy, a negative ATP consumption, which means that, yeah, they could run, but they cannot sustain life. Those that have zero will also not be able to sustain life. So an organism will not be able to live by doing exclusively this reaction. And then the ones from which we find the highest ATP yields or energy yields will be able to sustain life. Even if you have one proton translocation in net, you will be able to sustain life because you do it many times. And then we take those as the possible ones. And then you can postulate that the ones with the highest ATP yield should be, but by natural selection, should be the ones that are most favored. And this is, again, where we threw in the hypothesis here. We use an algorithm for this. I'm not going to go through it. So this is done automatically for each one of the reactions. And then we played with a number of scenarios, because obviously you're going to tell me, yeah, but we have a lot of parameters here that you need to put values to them. Well, we have some parameters, but not that many. So the only things we really needed to put values for are in the table here. The first four, actually, physiological parameters we needed to decide on. We need to put a number for our energy quantum for ATP hydrolysis, which is normally around 50. So we use 50, but we tried also a little bit smaller or bigger value to see whether this is having an impact, because we know different values have been reported in literature for that. We also tried different stoichiometries for the proton to ATP in the ATP synthase. So around 3 is a typical value, but we have seen papers talking about 15 for every turn. Protons, I mean, the ATP synthase makes 3 ATPs per turn, I think. So how many protons you need is divided by 3. It gives you the number of ATP protons per ATP between 3 and 15 over 3. We cover that range. We needed to give a value to some internal metabolite to be able to calculate things, because yeah, we're calculating things with this Gibbs formula that I showed you before. So products divided by substrates. So we need to have concentrations for everybody. If a reaction is insidious, it's easy because I have my initial substrate coming in, and then I just calculate for Gibbs 0 what needs to be the concentration of my product, and then the next one goes the same way, and the next one goes the same way. But for free CoA, enzyme A, we have to give a value to it, so we put the number in millimolar, and we cover the range as well. And we wanted to play with pH inside the cell, because it has implications in terms of the energetics as well of some of the reactions. So these are physiological parameters we play with. And then we also wanted to see if the results change, whether the results change at different environmental conditions. So what if the hydrogen concentration or pressure is different, right? So we saw before how that has a huge impact on the total available energy. But we wanted to see also whether it has an impact on which reactions go and so on. So we cover a range from 0.01 Pascal to 316. CO2 is also a product, same thing, we cover some range. We played even with temperature as well. And we applied the Van Hoff equation to correct some of the reactions. So actually temperature is very relevant here. And even the outside pH. Remember, we are dealing with a reaction which is incredibly close to equilibrium. So it's almost no energy here. So even a small change in temperature, if it changes some of the Gibbs energies, it might actually make the reaction feasible. And other reactions that were not feasible might actually run. So we wanted to make sure we covered those combinations. We tried combinations, and then when you start playing with these variations of parameters, you jump from the 500,000 options to 18 million combinations of options and sets of parameters that we wanted to play with. And we ran some experiments at a particular we run some cases at particular concentrations. So for example, for methanogenic conditions, typical values we picked from a reference source, from a standard kind of default typical conditions of propionate 0.14 millimolar and acetate 3 millimolar, and then we played with these guys. Okay, so what kind of outputs this is going to give us? What we wanted with this work is, okay, tell me which reactions are actually running and which ones are running with a positive net yield of ATP, which means they can actually sustain life. And we grouped these reactions. So the P1, the number one is the big family of reactions with which they belong. And the little letter was the secondary level, which was the electron carrier combinations. And then we don't have, or we cannot plot at the same time all the configurations of protons and so on. But we had already first results in this graph. What are we looking in this graph? So it's how much ATP per mole of propionate you obtain, and how much was the actually energy available in that particular reaction, which is the hat here, the black one. So in this case, this doesn't change. And then here, the bars are telling me which reactions have how much yield of ATP in these conditions of the table, right? We are using now the values 50 for the energy quantum of ATP, 10/3 ATP synthesis stoichiometry, these values for concentrations, and these values for propionate and acetate, which are, the propionate is the substrate and the acetate is one of the products. CO2 is given here and hydrogen is given here. So we can actually calculate the energetics like we did before, which is the thing here, this line, around 33. And then out of that 33, how much ATP can we make, right? So the idea is how big are these bars, right? So we have, in this particular example, numerical example, in this particular case, just to show you the kind of results we get, the lactate pathway was actually two pathways in the lactate family were the ones giving us almost half, I think, ATP per mole of propionate. The others were giving us less fractions of it. Okay. And then, of course, we can just start pushing for many more results. So now we can play with the, for example, for different values, how is the size of the ATP, the amount of energy in ATP? If we play with that, we see some differences, right? Some reactions disappear and some reactions appear. What if the ATP synthase has a different stoichiometry? We can see how the ATP, how much ATP is produced in each one of them. I'm not going to go into the discussion of results. I mean, one thing that is nice is that we can have a visualization of the efficiency in the sense that when a bar gets close to this line here, the line shows you the entire amount of energy available, right? The bars show you how much energy we are actually getting as ATP. So how much energy is the cell harvesting as ATP, right? So the closer we are, the bars to the line, is the more efficient actually pathway. The more efficient is the metabolism. That's another piece of information we get from here. And then we played with also different values of the CoA and different values for the pH side. I'm not going to really go into the results. So now if we change the environmental conditions, for example, if we change the hydrogen, we know already that the energy available is going to change. We calculated it before. So that you see the black lines are different. As we decrease the hydrogen, there is more energy available because the hydrogen is a product, right? So the Gibbs energy will be more negative, more energy available in the catabolism. And then if there's more energy, there will be more pathways that are going to be able to sustain life, and they will show up in this here. If we increase the hydrogen pressure, you see how difficult things get. This one is the Smith-Thella pathway, which is a slightly different stoichiometry, the number 7. That's why it has a different value. Okay, we play with other variables. Even temperature has an impact. So the higher the temperature, the more favorable is this propionate oxidation. So that's also how you analyze the effect. And the nice thing is that we can also make plots about what's happening inside. So let me say I pick a pathway, one of the ones that gives me some net ATP, and I can investigate what's happening inside. So if I have propionate, remember to acetate, CO2, and hydrogen, and this is a lactate pathway. So we go from propionate outside the cell at this concentration. This is logarithm 10, so 10 to the minus 4. I start here, and then I go to propionate inside, okay? And then propionate CoA, and then acryloyl CoA, lactoyl CoA. So this is basically the optimum arrangement of concentrations inside the cell. And then you see here, protons translocated. So here we are investing, so we are paying price to get the reaction running here as well, but then you get payback here. So you get another proton back here, and you get an ATP SLP here from the CoA, the activation of the acetyl CoA, and then another one here. And these are for 3 different values. They're actually 3 in 1 for different concentrations of hydrogen. Okay, so the low one, the triangle, is the at the lowest hydrogen pressure, and you get a net ATP when you count all these bars of 0.70, and at the highest one we get a 0.10 net ATP if we follow this. So this actually gives us a lot of insight in what's happening. And at the bottom, I wanted to plot the losses, right, the inefficiencies. So if a reaction is going, I mean, we cannot increase further the concentration of the product because we cannot exceed the ranges, right, of 10 to the minus 2 and 10 to the minus 6. So a reaction, if it's high energy in it, you might want to actually have a higher concentration of the product so you don't lose the energy and you use that high concentration to make the next reaction easier. But sometimes you cannot, because you cannot exceed those ranges that I showed you. Also, you cannot go lower than this value over here. So sometimes you need to lose energy, and that means you lost that energy and you didn't harvest it. So when this happens, you cannot harvest that energy, so you lose efficiency in the system. And this is where you lost it, are these yellow bars. All right. So that was a propionate oxidation. And then quickly, what we did is we coupled this to hydrogenotrophic methanogenesis. Hydrogenotrophic methanogenesis is CO2 plus hydrogen making methane. It has a known stoichiometry, so we did the same thing. In this case, it's only one main pathway, but we created variations of it only in terms of the number of protons that could be translocated in certain steps. Okay. So we did that, and now what we did is we ran them together. Okay. So if we have a scenario together, what are we looking at? This is a huge graph, which is quite scary. Is at the top is the same thing you saw before, is the propionate oxidation itself. And what are we looking is at different pressures of hydrogen, right? So which is here. It's the same axis in different units. So hydrogen pressure. So if we move to the right, there's more hydrogen in the system, which if you remember, it was making more difficult the reaction to run for propionate oxidation. So the little lines are going closer to zero. And these bars are the options we have to do the pathways that are going to be having positive ATP yielding for the cells that are doing those works, right? And then we do the same thing for the middle one is the methanogenesis, right? So it's the one consuming the hydrogen into methane, hydrogen and CO2 into methane. And obviously for low hydrogen pressures, the reaction doesn't run. There's not a single option that runs, but then from a certain value of 1.4 Pascal, then yes, there is the pathway can run. We have a first bar here, and then it becomes easier and there's more energy available. So the more hydrogen there, the less energy for the first one, and the more energy for the second, because in one is a product and the other one is the substrate. So these guys live together. So they need to live in a range in which both of them can have bars showing net ATP yields, right? So we could compute this in these ranges. This is for a scenario in a very favorable situation, which is very unrealistic, with a very high propionate and low, so high propionate and low acetate concentration, which makes things easier. And we have this kind of situation in which we have a broad range. The green area here is where both can live and sustain life between 1.4 Pascal and then 11,000, 12,000 Pascal. Right? This is a scenario that never happens. We normally are in a situation where we live in methanogenic conditions in which the hydrogen pressure is, I mean, the concentrations are these values here that are both small. And then when we look at the energy available, we have a narrow, super narrow range of hydrogen or theoretical hydrogen concentration in which both of them could live. When we calculated that, what we found is that the value was 10 to the minus 8. And 10 to the minus 8 is forbidden because you remember we couldn't have things lower than 10 to the minus 6. 10 to the minus 8, it means you have virtually nothing inside the cell. So one of the conclusions that this work tells us is that it must be something else than hydrogen. Maybe other things that are not hydrogen are actually doing the electron transfer between the two syntrophs. Things like, for example, formic acid has been postulated. If we calculate the equivalent formic acid energetically, then we have values in the order of 10 to the minus 6. So this could be possible. We could also do that via electrons directly, electrons at certain voltages. Okay? And that would place this area in 270, minus 270 millivolts, for example. In this, the electrons could, the level of energy of the electrons could be feasible for both of them to have enough energy in the propionate oxidation and have enough energy on the methanogen. All right, and that's pretty much the same thing. So just to conclude, what we try to do here with the talk is basically to, the message I want to convey to you is to, encourage you to use thermodynamic calculations, because sometimes you can get an actual insight of what biology can do, especially in energy limited systems. If you have systems that are energy constrained, in which they're normally anaerobic systems, these can help you calculate at least the things that can never happen. Okay, obviously, if a reaction can happen theoretically, it doesn't mean it will happen, because it might not be just anybody to do it, or maybe other limitations, kinetic limitations, but it can actually constrain or just remove areas of impossibility, and then we don't need to worry about it. Right? It's a good tool for that. And let's not forget that the living things must also follow the laws of thermodynamics. So there's no way around this Gibbs energy story. So you cannot have a reaction running with a positive Gibbs energy. Then energy conservation is another thing, right? The energy conservation in energy tight systems, it has to be very efficient. When we are very efficient, it means the metabolism is not losing almost any energy. If you don't lose any energy, it means this delta G that we are having in every step needs to be tiny, very close to zero. And then if it's close to zero, it's close to equilibrium. So these guys are almost running very close to equilibrium. And the good news is that we can calculate the equilibrium. We know equilibrium because of the thermodynamic properties, so we can calculate where it is, and these guys are close to it. So it might tell us a little bit about what they might be doing. That's one in the first part. And then in the second part, well, it's just wanted to show you the method we developed, which allowed us to actually evaluate combinations of pathways virtually for any microbial conversion. Now, we created the algorithm in a way that is general, so we can actually bring in, if I don't know, a different reaction. Any process that is energy limited, we can just put it in. We create all these families of pathways, combinations, options. We threw it in, and it will generate these outputs for us in terms of which pathways are feasible, which pathways gives us how much ATP, and so on. And we can even look inside the pathway and look for bottlenecks of these intermediate concentrations that I was showing you. If we are getting into a super low limit, it means there's a bottleneck there. Maybe we can do something about it experimentally. In the case of propionate oxidation, we found the lactate pathway as the most energy yielding. And then another conclusion of this theoretical work was that in syntrophic propionate oxidation with methanogens, it looks like hydrogen is a tough one to be the guy that is being the actual interspecies electron transfer mechanism. It's probably going to have to be something else, which could be formate or other electron carriers, or even direct electron transfer through some sort of contact, or physical contact, or membrane, protein, or something like that. That was the conclusion of the work. And that's all I wanted to show you today. I think, yeah, I hope I did okay with time, and then we have some time as well now for questions. Thanks.
Thank you, Jorge. This is really cool. Yeah, we do have some time for questions. Maybe I'll start. While if anyone has questions, either type it in the chat or in the Q&A, or raise your hand and I can unmute you. I'll just start by asking kind of two related questions, if you will. So in metabolic pathways, right, a lot of reactions are unidirectional, but some are bidirectional. And I was wondering if this is something you kind of accounted for, or you think about accounting for. And maybe a related thing is, so you're doing all this, I think, assuming that everything is going to steady state, like, right? You're doing thermodynamic calculations, but is kinetics important at all? Does it matter if how fast the reaction goes, and the bottlenecks of different reactions have an effect on any of these things?
Yeah, definitely. So the reversibility, yes, absolutely. Most of these reactions are reversible. The direction in which they go is dictated by, well, if they are reversible, is dictated by the Gibbs energy. So if the Gibbs energy is negative or positive, it means it's negative in the other side, in the other direction, or the other way around, right? If something is negative Gibbs going to the right, then it means it's positive Gibbs going to the left. So this is clear. In this case, we studied the propionate oxidation in a situation in the system in which it's favorable to go to the right, as I presented it, right? So we calculated all the steps to harvest the most possible energy. So the point of this is to find what biology could have developed to harvest the most possible amount of energy in terms of evolution. So the underlying hypothesis here is that whatever is the most effective, it favored growth, and therefore it favored selective pressure towards that. So we are looking into studying that thing. Since it's steady state, there is no reversibility or not. So things are actually running in one direction. in this moment. But of course, we could change the system. We could increase the acetate concentration and then do acetate to propionate. And then if it's going in that direction, we evaluate it in the opposite direction. So in that way, is there. That links a little bit with the kinetics question you put in there. As I was saying, the thermo will tell us what can happen, but it's not going to tell us whether this is going to happen or is going to happen at a meaningful rate. Obviously, I mean, in this case, what we did is we picked the inventory of reactions which are known to have biochemistry to run them. So each one of those steps, they're well known. So they are almost central metabolism steps. They are known, the enzymes, there are enzymes that do them. So they are all well characterized. And then it will have, I mean, there will be a rate, of course, per unit of microorganism, but that rate will not affect the yield in this sense. So if you're limited by substrate, everybody is running at the same rate. So the guy that has most energy harvest capacity is going to have a higher yield and therefore a higher mu of specific growth rate. So the mu will be the product of your yield times your rate, and then you have this yield rate, dichotomy. But if you're limited by substrate, everyone is in the same terms of rate, how quickly you can uptake the substrate, then it's going to be the yield what makes the difference. If you're more efficient, you're making more business out of that propionate than the other guy, then you're going to be growing more. And the hypothesis is that will be favoring your selection in the ecosystem. And this is kind of the underlying.
Pretty cool. I wonder if you can use this kind of method for other metabolic pathways to kind of evaluate, could certain, metabolic pathways exist on Mars under some type of condition?
This is definitely one of the things I really wanted to start doing immediately is, well, first from the process development point of view, there's a lot of processes for bioenergy and to fight climate change. There are a lot of people looking into how to convert CO2 into fuels and all these kind of things. So those reactions are going to be, all of them, very low energy in terms of low energy consumption, because you don't want to burn the energy, right? So you want to actually keep the energy in your products and your fuels. So they are going to be perfect for this kind of configuration. And then, yes, astrobiology will be really, really nice. So this is one of the things I always wanted to put my nose into. We can literally look into any chemistry. The only thing we're going to need is to have an inventory of possible biochemistry that can do that, right? Or, of course, we can postulate that if this would be possible, then we just imagine we put one of those reactions, imaginary reactions there, and we can still do theoretical calculations in terms of what could be happening and what could not be happening in terms of thermodynamics. Yeah, that would be really nice. Someone wants to start a collaboration on this, well, please let me know. I will be very, very excited about it.
Any questions? I think if there's no one, I guess we'll just end it here. And thank you, Jorge, very much for a really cool work. And hope to see you around Abu Dhabi sometime.
Yeah, thank you for inviting me, guys. And yeah, I'll try to keep up with the rest of the seminars. It actually looks a nice program that you're running, so I'll be joining some.
All right, good to see you.
Thank you.
Bye.
See you.
Bye, everyone.